Hello, good afternoon everyone. Welcome to the Natural History Museum and welcome to Nature Live. My name is Camilla and I'll be your host for this afternoon's event. Now for those of you who haven't come to a Nature Live talk before, I'll give you a very brief overview as to what we're about. We host these talks to give all of you the chance to see some behind the scenes stuff, usually at the museum. Today's is slightly different, but what we normally do is we bring out some of our 80 million strong specimen collections here at the museum and we introduce you to some of our museum scientists. So it's your chance to see some cool specimens and speak to a scientist face to face. You can still speak to a very accomplished scientist face to face today, but he isn't based here at the museum. So he looks after a bioimaging unit at Oxford Brookes University. His name's Chris Hawes and we're very lucky to be joined by him today. If you've got any questions for him throughout the event, just pop up your hand and he will do his best to answer them. So Chris, thank you very much for joining us this afternoon.
Fine.
Tell us a bit about what you do over at Oxford Brookes.
Okay, before we start, there's obviously a few school groups here, yeah? Anyone doing biology? Yeah? Put your hands up if you're doing biology. So there'll be some words I expect you to understand in the next half hour then, that's great. So you can tell the person next to you if they're not a biologist. Anyway, yeah, I'm a professor of plant cell biology at Oxford Brookes and I run all the big microscopes we've got. I've just loved plants all my life, but when I was at school we picked up microscopes for the first time and that was it. Once you've used a microscope you will never want to use any other instrument. So hopefully at the end of the afternoon, especially the younger ones of you, will want to go back to school and demand to use your microscopes. So we're going to have a tour inside plant cells for the next half hour and see what we can see.
So we're going to look at some of the incredibly advanced technology that you have over at Oxford Brookes in a moment. But why don't we begin by finding out why is it that you think plants are important to study? Because a lot of visitors to the museum are excited to see the dinosaurs and the mammals and stuff, but why do you feel it's important to study plants?
Yeah, all our students hate plants. They want to work on fluffy animals and things like that. But if you think about it, if we can have the first slides, everything you eat comes from plants. Everything. Even though we might feed it to a cow first, like grass, and then you eat the cow, it still comes from plants. So it's the start of the food chain. All our energy comes from plants, via sunlight, photosynthesis, making oil, making coal, making wood we burn, all the biofuels we're now making. Eventually solar panels will be based on leaves, not on the chemical systems we're using nowadays. And loads of drugs come from plants. Something like 80% of the world's population rely on plants for their medicines. Herbal medicines aren't rubbish. Aspirin, which you've all heard of, comes from the bark of willow. It's now synthesized, but it came from willow bark. So the importance of plants cannot be underestimated. But if we look at the next slide, if you look at that graph up there, we're going to get to about a population of 9 billion in the world in 2050, which means we're going to have to increase our agricultural production by another 100%. So we've got to produce twice as much food in the next 30 years, 35 years, and the amount of land that we can grow crops on is rapidly shrinking because of global warming. Deserts are taking over, not all, but a lot of farmland's getting salty. You can't grow the same crops. So we've really got to work on plants and plant science to try and up our game so we can actually feed ourselves if we want the kind of civilization we've got today. It's as simple as that. We haven't got an option. So plants are the cool things to work on at the moment.
Okay, you stated a pretty good case there. So this talk is related to a very important historical figure in science and microscopy. Has anyone, put up your hand if you've heard of Robert Hooke? Hopefully a few of you.
That's great.
Good, okay,
Good,
so
good.
Can you tell us a little bit about him?
Yeah. Hooke was an amazing guy. He was what we call a polymath. He could work on anything. The good thing about Hooke is that he came from Oxford. The bad thing is that he came from Oxford University, and I'm from Oxford Brookes University, which is the modern thrusting university in Oxford. But we do talk to the guys down the road occasionally. But I think Hooke went to Wadham College, and people don't realise this guy Robert Hooke, he redesigned London after the Great Fire, but they wouldn't accept his designs. He wanted a grid system like New York. This was back in the 1600s. And he was too advanced, so they actually rebuilt London on its old pattern of curved roads and everything. But he was an architect. He worked with Christopher Wren on redesigning and rebuilding London after the Great Fire. He worked with Robert Boyle. Do you remember Boyle's Law? Do people remember that, on gases and things? He was a chemist as well. But the important thing was, and in those days it was sort of an amateur science, he liked microscopes. And if we have, oh yeah, his microscope's up there. Can we have the next slide? Is that a bigger one of it? Yeah. So this is the kind of microscope Hooke worked on, and he just drew whatever he could see down a microscope. They were pretty crude instruments. We're much better off nowadays, as you'll see. But he drew fleas, he drew flies' heads, and people are actually saying, well, his microscope, he couldn't have seen all of that. There's a bit of imagination going on there. But the next one is even better, I think, the fly's head. And these are the kind of things we see in Sunday Times magazines from scanning electron microscopes and everything. But if we have the next one, which is a much more simpler diagram, the cork one, yeah. There's a piece of cork that he'd sectioned, and he saw all these structures in there that are round. And different people have different stories about this guy. And some say, well, he said that looked like the cells in honeycomb. Other people said he thought it looked like the cells that monks used in abbeys and things like that, where they used to go and pray. And so he termed these structures cells. So that's where the word cell comes from. So Robert Hooke coined the term cell, and for microscopists that was the key thing. And he published all these pictures in the first ever publication from the Royal Society 350 years ago this year. So it's the 350-year anniversary of that Micrographia magazine, if we go back a couple of slides.
So this was 350 years ago, and actually at that time this was quite a revelation, because all his drawings, this was the first time society was seeing these tiny things in such
he was miles ahead of his time, miles ahead. We didn't even have the equations to tell us how a microscope worked. They were just put together with lenses to see what happened. Nowadays we know exactly how they're working. We can get much better resolution. We don't talk about magnification. He would have talked about making things bigger. We talk about what we can see in a microscope, and that's the resolution. You can blow an image up to any size you like nowadays with projectors. Doesn't matter. It's what you can see. And so what we're going to do is get inside cells, basically. We're going to have a look inside leaves.
So you've seen he's drawn pictures of insects and things like that. He drew lots of things. He drew urine. He drew the edge of a razor. He drew lots of different things. But he was also very interested in plants. So I'm going to read you a very, very short extract. So he experimented with plants, and he cut into a stem, and he noticed this liquid. So he said 2 or 3 drops of liquor were thrust out of the part from whence that was cut, very clear, of a bright greenish colour, tasting at first a little bitterish, but after leaving a licorice-like taste in my mouth. So he actually tasted it. And he also referred to the visible canals that this liquid travelled through. So do we now know exactly what he was talking about?
Yeah, you probably heard, those of you at school doing biology have heard of vascular tissue in plants. The conducting tissue, the long cells that go up and down a stem and down a root. And there's one lot that takes water up to the leaves for photosynthesis. And there's another set of tubes that will take all the sugars and everything that are produced by photosynthesis down to the growing parts of the root tip, or the meristem at the top of the shoot, so the plant can grow. I'm going to show you a video of the cells that transport this sticky, sweety fluid that Hooke tasted using one of our new microscopes. I'm not going to tell you how we did it, but just use the stereo glasses, red on the left, and see what you can see. And we'll go on a trip just down one of these tubes that Hooke thought he could see.
Okay, so we play video number 5 for you all. Maybe dim the lights if possible.
I have the flashy glasses, the better one. So we're actually inside a cell that's conducting sugar throughout the plant. And that tube would be about 10 microns in diameter, and a micron is a millionth of a meter, to give you an idea.
So this is one of those canals that he mentioned. What are these tiny little blobs that we can see as we're travelling down?
The little blobs are structures called mitochondria that half of you would have heard of. Yep. All the white stuff around the side of the cell is a structure called endoplasmic reticulum, which some of you would have heard of. That's where the protein's made in cells. And we're going to see more of that in a minute or two.
So another plant that Robert Hooke was very interested in was nettles. So he would sting himself multiple times on the leaves and try and understand what was causing this pain. He's got this beautiful drawing of nettle leaf here. But today we can produce images like this using something called a scanning electron microscope. Oh, yes.
And also, Camilla wouldn't wear this, unfortunately. We did a conference in Oxford 20 years ago, and to celebrate that picture of the nettle that Hooke did, we made a T-shirt of it. So if anyone wants to look at the T-shirt afterwards, you're welcome. That's a very, very famous, the first ever drawing of stinging hairs on a nettle.
So talk us through a little bit about how a scanning electron microscope can produce an image like this.
So you might have all seen pictures like this in the Sunday Times magazine or the Observer magazine of fly eyes and everything. So what we do, this is the surface of a leaf. These are the stinging hairs on the nettle. And the scanning electron microscope has a beam of electrons, and it scans it backwards and forwards across the specimen like that. And electrons literally bounce off the surface. You collect them with a detector and turn them into a current, and that's turned into an image on a video screen. And these days it's all digitized and goes through computers. If anyone remembers the old-fashioned TVs with a cathode ray tube, which were under vacuum, they were shining a beam of electrons across the phosphor screen and building up the image. It's exactly the same as a scanning electron microscope. And if you think, if that was a whole leaf, and I just scanned a very small area, we'd get a bigger magnification. If I scanned a large area, we get a smaller, lower magnification. It's as simple as that. You get these beautiful 3D diagrams. So I think now we can go further into the leaf, I think, zooming.
Yeah, so hopefully we've got another clip to play you now, and that's put together by the head of our Imaging and Analysis Centre here at the museum. If we give it a couple of seconds, there we go.
And this is a tobacco leaf. It's suffered on the coach coming up from Oxford, I'm afraid, but this is one of the tobacco plants we work on. And we're just zooming onto the surface of the leaf.
So it's kind of hard for us to gauge a sense of scale here. Can you kind of describe that for us?
And that would be 20 microns across. And again, a micron is a millionth of a meter, so 20 times 1 millionth of a meter. So very, very small. So we're looking at the surface of the leaf. Now we're zooming down. And look at the structure of these cells on the outside of the leaf. These are what we call the epidermal cells. And we're going to get inside these cells in a minute. I don't know, can we put this on the visualizer?
If we bring up the visualizer, you've brought a little tobacco plant with you.
Oh, there it goes, that's great.
There you go. So the little dial above, you
So
can
I can
twist it
zoom on
and zoom
there.
in and out. There you go.
Oh, there it goes, it's electronic. Ah! You have to work backwards.
Push the little button on the end of the dial.
And focus it. Yeah. So what we just saw was all the cells on the surface of a tobacco leaf. But now we want to look inside the cells. And we're going to try and do it in 3D so we can model the whole cells. We're not going to use 3D glasses anymore, so you can not put those on.
So before we go on to do that, does anyone want to ask Chris any questions so far? Anything come to mind at all? Yep, go for it.
Oh. The materials guys, not the biologists, can get down to below the diameter of a hydrogen atom, which is unbelievably small. They can get down below what we call an angstrom unit, which is about the smallest you can think about, yeah. But what they're not looking at is they're not looking at the structure, they're looking at the distance between two structures. So you can get much finer resolution that way. But in biology, well, the virologists can get down to proteins on the surface of a virus particle quite easily nowadays, yeah.
Would anyone else like to ask Chris any questions before we move on? No, okay. So Chris, this machine, tell us about this, because to me that doesn't look terribly exciting, but this can do amazing things. So what's this?
This is our latest scanning electron microscope. It's quite a big beast. It can be used for taking those nice 3D pictures, but that's a bit of a waste of its capabilities. So what we've got inside the chamber here is a cutting machine, a microtome, like a bread slicer. So we can take specimens and cut them and look down into them. So if we look at the next image.
Should we show the video?
Yeah.
Yes, if we play video number 10, we can show people how this machine actually works, or at least an animation of how it works.
So what I've done is take a lump of tobacco leaf, we fix it with chemicals, dehydrate it, put it in a block of resin like household araldite, and then the beam of the scanning electron microscope scanning across and taking a picture. You can see it taking its picture there. And then we cut a section and throw the section away. We're not interested in it. And we just then scan the surface of the resin again. And that way we can work our way through the specimen slowly. At about 50 nanometer thick sections, which is very, very thin. A nanometer is a thousandth of a micron, which is a millionth of a meter. So we're getting thinner and thinner and thinner and thinner.
So this takes a long time, I'm assuming.
Yeah, this can be going for 2 or 3 days. It's great now doing microscopy. You set it up, you can go home and have a beer. Or actually, you can have 24 hours worth of beers and come back and collect your data. So that's okay. So we've done that. All that data is imaged and digitized and put in the computer, and then we can reconstruct it into a 3D volume and actually get inside a cell and see what it's all like. So if we have
let's have a look at video number 11. So this should be a lot of information combined and building a 3D image of the cell and the structures inside.
So this is the surface of the resin block showing cells in tobacco, and we're going through the specimen. And now we're reconstructing one of the structures. That's the nucleus in the cell. And now we're reconstructing the powerhouse of the cell around it, which are the mitochondria that we mentioned before. And then we're reconstructing the endoplasmic reticulum, which some of you heard about. This makes proteins in the cell, and this is what we're interested in our research. And then these little green blobs that are floating around everywhere, those are called Golgi bodies. A guy called Camillo Golgi in Italy in 1898 actually discovered these things at the light microscope level. They process proteins in your body, but they also make all the carbohydrates that you might secrete. If you've got a cold and you're making lots and lots of slime down your nose, these little green blobs that are in animal cells will make all that slime. In plants, they make complex sugars like the polysaccharides around the cell wall and everything like that. So they're very, very important for food processing, and that endoplasmic reticulum makes all the proteins. So all the protein you eat is made in that sort of it's a tubular structure really all around the cell. And we can see it in 3D now.
If we play video number 12, we can show proteins being made in a cell, hopefully.
So this is actually showing the endoplasmic reticulum where the proteins are made. So we're going through about a whole cell's depth in lots of thin slices. We reconstructed in 3D. There's the two nuclei again. And this red and the yellow, as it will come up, I think it's yellow. You'll see here. That's all the endoplasmic reticulum. And you could never see it like that before by electron microscopy. If you're seeing just one thin slice, you don't get an appreciation of what it's about. But now we can see it in 3D. We can actually measure it. We can measure its volume. We can do experiments on it. We can have the plants under different conditions. We can see when it's producing lots of protein, when it isn't. So it's opened up a new era of microscopy, just being able to do all that 3D work.
And you mentioned Golgi bodies. If we play video 13, maybe we can have a look at those as well. So just remind us what it is that the Golgi bodies do.
So the Golgi bodies are little stacks of membrane, about a micron in diameter, that make sugar and also process protein. And in our lab, they're our favorite structure in the cell. We've spent 15 years working on them now. But this is the first time we've ever been able to reconstruct them in 3D and see their position in the cell and then count them, the number in one cell compared to another. And it's just reopening up the world of the cell to us. And these microscopes have been around now for only about 4 or 5 years. So it's a very, very new technology.
So some of you will probably have heard of 3D printers, and you might have seen some of the models we have on the table here. Can you just talk us through these, Chris, and what we have in front of us?
Yeah, so if we look at the next video.
Yeah, if we have the 3D printer video, that'd be great.
So we've got a project at Brookes going on that we thought, yeah, you guys can see all that, can't you? It's quite easy. But there's a lot of partially sighted people out there, or blind people, who've got no idea of microscopy at all. If you can't see well, you can't use a microscope. If we can now print out the structures in a cell, we can just pass these around. That's a 3D model halfway being produced, this one here. And you see all that, there's a filamentous network we have to pull away, but we can actually reconstruct the structures in the cell. That's the nucleus I showed you. That was the blue blobs. That's where all the DNA in the cell is. That's one of those Golgi bodies, so we've made it green. Some other things just to interest people. There's a virus for you. And this is one we reconstructed. So totally different. If you were infected by that, you would not be healthy. This is even worse. Nothing to do with my talk at all, really, but it's just a nice model. This is a trypanosome, African sleeping sickness, that we reconstructed using the same technique. So and you can imagine, if you haven't got good eyesight, you can actually feel the structures at the microscopic level. And we've never been able to show people that before. So it's opening up microscopy to a whole new range of people.
And you've conducted outreach with these models with partially sighted people,
Yeah,
haven't you?
yeah. We've taken them round to groups of partially sighted people. You actually take a microscope as well, and people can feel all the parts of the microscope. And even though it's just a small one like you might have at school, it's quite easy to extrapolate that to one of these larger microscopes.
If we could have those back actually before the
Yeah,
end.
don't steal them.
So a bit of a random question, but is it problematic to only work on dead things? Do you want to work on living things instead?
This is the problem. Everything I've shown you so far is dead. It has to be dead to do that kind of processing. An electron microscope's under vacuum. So if you put any biological material under vacuum, it'll just collapse totally and die. So we have to preserve stuff by fixing it and embedding it in resin. And that's a problem. So now we've got more advanced light microscopes where you can look at living material and look at the same structures in the living situation rather than the dead situation. Not at such high resolution, but when you can see a cell in action, alive, it's a revelation. So if we continue on to how we do this, I know I need my pointer.
So first of all, tell us what we're looking at here in this image.
So, back in the late 1950s, a guy called Osamu Shimomura from Japan, a PhD student, worked on what we call bioluminescence. Everyone heard of glowworms and fireflies and stuff like that? They make their light by chemicals. But there is a certain number of organisms that do what we call fluoresce. They will absorb light of one wavelength and give light off of another wavelength. And you have to have specialized proteins or chemicals that form ring-type structures called fluorophores to do that. And there's this brilliant jellyfish in the North Pacific, called Aquaria victoria, that if you shine UV or blue light on it, will glow green. And this guy Shimomura discovered this protein in the 50s, and he got a Nobel Prize for it about 6 or 7 years ago.
Didn't he discover it kind of by accident?
Yeah. So this protein, he was desperately trying to extract it, but failed. And he was so frustrated in his lab one night, and this was on the west coast of the States, he chucked a bucket full of extract down the sink. And there was salt water in the sink, some seawater, and a big flash of light came out. And he thought, oh, something's going on here, obviously. So he worked and worked for another few years and discovered one protein that would give off a blue light in the presence of calcium, which you find in seawater. And that blue light he discovered was what made the jellyfish fluoresce green. So he found this other protein, and being inventive, he called it green fluorescent protein. And he got the Nobel Prize for it. And no one knew what use it was at all, 30 years before there was any use for it. But this is it there in his pot. And
Chris, actually, I've just had notes that if we leave the bottle just there, we've got a nice shot of it, but we can ask someone to shine.
Yeah.
Is that okay?
So is anyone good at aiming lasers? Volunteer. Okay. Promise me you won't drill it in the back of the heads of the people in front of you. There's a little button there and there's a blue laser. Now, can I turn this visualizer off? Yeah. No, it's not going to go off.
That's okay. It's nice and dark.
Try and hit this little jar of green fluorescent
pretty good
protein
aim.
there. Oh, nearly. Oh!
There you go. So what's
See what
happening?
happens?
What's happening here?
So what the chemical in there, it's a protein that forms a circle. It's absorbing the blue light, and we say getting excited, and it absorbs the energy, then the energy decays, and it gives off photons of light in the green. So this protein is just absolutely brilliant. It's even better because some guys cloned the gene from it. Can I have my laser back? That was a good shot, by the way. So some guys in the States actually cloned the DNA that makes that protein and discovered that if you put that in bacteria, you could make them glow green. If you put it in worms, they'd glow green. You put it in plants, they'd glow green. So back in the 90s, we put it... Oh, that's the wrong... Oh, there's my other one. We put it in tobacco plants. Well, we put it in various plants. And there's a tobacco plant glowing green with this protein on it. That's under ultraviolet light. It's exactly the same as one of these, but that's got a foreign gene in it. This one hasn't.
So if
So-
we play, sorry,
Sorry,
continue.
no.
So say if we play maybe video 19, and you can talk us about the uses of using this.
So what we did, we took that protein, and there's various variants of it. We've got one that will fluoresce red, and there's one that will fluoresce blue as well. And you can take the gene for that protein and actually join it using genetic engineering to a gene for any protein you want that you know that's in your cell. So we took a small protein that would be in the endoplasmic reticulum and attached the green fluorescent protein to it. And we took another protein that we knew that would be in the Golgi body, those little green things I was showing you, and attached a red fluorescent protein to it. And if you see in this video here, you see all the green, and there's little sheets and tubules. They're very small, but there's even smaller red blobs around. Well, those are the Golgi bodies, and the green is the endoplasmic reticulum. These big cylindrical things, those are actually what we call defense compounds. And in cabbages, that's what makes cabbages smell when you boil them. And they're anti-insect compounds in big crystals, and they absorb this protein we've been making.
So let's look at video 20, and we can see a higher magnification actually.
So here you are, that's one cell in the leaf. It's exactly the same as the cells we were reconstructing with that scanning microscope and doing the 3D reconstruction. This is speeded up about 2 times. So the first thing you see is everything's moving. It's really, really dynamic. That's the leaf, that's what's going on inside here. So every leaf you see out there, it's not just a plant being static doing nothing. Inside the cells, they're really, really dynamic. And all that movement is generated by exactly the same proteins that you've got in your muscles that make us move. Exactly the same. 10% of the protein in a leaf is what we call actin, which is the meat in your muscle. So plants are using the same motile systems, but inside the cell, rather than for moving the whole plant around.
And this actually showed you more clearly that the Golgi bodies and the endoplasmic reticulum are much more sort of connected than we first thought.
Yeah, this will really bore you. What we're interested in is how material gets from that endoplasmic reticulum into the Golgi to the outside of the plant, or to where it's stored in the plant. And we thought those two structures were joined together. No one else believed that. But you look at those movies, they look as though they're joined together, don't they? Because the Golgi are actually moving, those little red dots are moving over those green tubules, and everything's moving. So we thought, well, how could you test that physically? And actually, there's a great way with lasers. I mean, it's the last movie, but this is a green laser. You can make lasers of different wavelengths. They just have one single wavelength. So you can have an infrared laser. Infrared lasers are really weird, because if you shine them into any solution or material with particles in it, the particles are about a micron in diameter. They get sucked into the laser beam. It's sort of complicated physics, but we call it laser tweezers. So you can grab something.
Let's show the final video. Let's show video 22, and then we can watch that while you're talking about it.
So what we're doing, we're using a laser beam. You can't see it, but we're actually going to grab one of these Golgi bodies, the red ones. We know we actually can't grab the green structures. And we move it around the cell. It's a little bit distorted here, but it doesn't matter.
So that movement is being controlled by someone controlling a laser?
Yeah, we're just moving a mouse around controlling a laser. And can you see that the tubules are being reworked throughout the whole of the cell? And this is only a small part of one cell. So we can actually reconfigure the inside of a cell. Okay, there's a bar there, 5 microns. A micron's a millionth of a meter, thousandth of a millimeter. So 5,000ths of a millimeter, yeah. Pretty small. And because we're just pulling the red blobs around, if we stretch the green tubules, we know they're attached together. We know they're physically attached. And that upsets a lot of people. There's a big argument. That's why science is great, because you can have these great arguments. And we had a brilliant, one of my PhD students this week only came to me with a brilliant result, that what she'd done was, we've got two proteins we think join those two structures together. So she's mutated those 2 proteins and put them into the tobacco system here. And what she's found, by upsetting those 2 proteins, she's stopped transport of protein from the tubular endoplasmic reticulum to the Golgi. So we think if you break that link, you break transport. So we've upset the transport pathway. So that's telling us how material is moved around the cell before it's sent to where it's going to be stored so we can eat it. So we're slowly dissecting the pathway. It's like going into the Mini factory, the BMW factory at Oxford, not knowing how it worked, and then slowly taking it apart to work out how it works. That's what we're doing at the microscopic level.
Okay, so we are very much out of time, but does anyone have any final questions for Chris? We can take one if anyone would like to ask him anything. I'll ask you if we can be very brief, Chris, but I'll just quickly ask you, this technology is obviously amazing, and it means we can understand plant life in a whole new light. What's your kind of vision for the future? How's this going to affect, I don't really like using the word benefit, but how's it going to affect or change the way we think about plants for the future?
Hopefully we'll be able to manipulate plants and make them grow more efficiently without just doing blanket genetic engineering. At the moment, we try and change genes and see what happens. If we fully understand how the system works, then we'll be able to target specific parts to make it more efficient. That's our ultimate aim in our lab. Other labs have different approaches, obviously, but this is just one bit of a complete complicated structure. There are many, many other structures in a cell that people need to work on as well, and they indeed are working on. So what you're seeing there is accumulation of 15, 20 years work in our lab and lots of different people, and we're still nowhere near actually understanding exactly how it works. But that's why we do it. It's great fun, it's interesting, and hopefully it'll help mankind as well.
And you can have a few beers while the SEM's running,
yeah.
obviously.
We can go home while the experiments are running, and that's brilliant.
So guys, we are just about out of time. Thank you very much for coming. Thank you for your wonderful questions, but please do join me in thanking Chris.